Okay, so here we are. My name is Rex Hauser, and this is my presentation on the influenza virus, also known as Orthomyxoviridae influenza virus. And this presentation was originally to be done on 14 May 2018. So here we go. The influenza virus. And I broke it down into many categories. So, first category, the etiologic agent is Orthomyxoviridae influenza virus. There are three influenza virus types, A, B, and C. Type A has subtypes, and they're based on the surface proteins and the two surface proteins. One is hemagglutinin, and the other is neuraminidase. And presently, there are the H, the hemagglutinin surface protein. There are 1 to 16. There are 16 variations of them. And on the neuraminidase, there are 1 to 12. And they're still finding new ones, ones that have already existed. I guess variations. It's still mutating. So those are the types that are in circulation right now. Few type A subtypes are in the human population, like H1N1 and H3N2. Most are in other animals, may cause epidemics and pandemics. Type B, no subtype classifications for type B, causes illness only in human population, may cause epidemics but not pandemics. And then there's type C, which causes only mild illness and does not cause epidemics. My subcategory is type A subtypes. We'll go into there. Here is a display of the different H and N glycoproteins and how they combined and in which animal types you find them in. And as you can see there, the humans are on the far left side of the graph on both of those, on the H and the N side. And you can see that there's combinations that are found in human beings, in other animals, and there's a lot of crossover. And that's, I guess, the main takeaway from this, is there's a lot of crossover in these types. And that's actually one of the virulence factors we'll get into in a moment. Reservoirs. It's mostly in birds, mostly wild waterfowl, and in pigs and swine. And the hosts are many other animals and humans. And my understanding is the difference between reservoirs and hosts are the reservoirs carry it, and for the most part it doesn't cause them much illness. And hosts are mostly intermediate, and it can very well cause them illness. So I have a sub-topic here, reservoirs and hosts. And as you can see, the influenza virus basically comes from the waterfowl. And then it spreads over many other animals, including humans. And as you can see there's more interplay between the waterfowl, pigs, and human beings. And this becomes very important in a moment when I discuss the virulence factors, because the contributions, the new mutation contributions of the waterfowl and the contributions of the pigs or swine and human beings mix and match and create new viral strains. And these can be dangerous. These can cause pandemics. The body, the human body, doesn't have resilience against these yet. It hasn't built up. So when these develop, they could be like a wildfire and go right through a population, and then you have a epidemic or a pandemic. So I'll discuss that in a moment. Morphology. The virion size is 80 to 120 nanometers in diameter. The shape is pleomorphic. It runs from spherical to filamentous. The type is enveloped with an 8-segment, single-stranded, negative-sense RNA genome. It possesses glycoprotein spikes, and these are those surface glycoproteins I mentioned earlier. The HA stands for the hemagglutinin. There are 500 of those per virus on the surface, and the NA stands for neuraminidase, and there's 100 of those spikes per virus on the surface. Also included in the virus package, so to speak, are the matrix proteins, designated M1, integral membrane proteins, designated M2, RNA, and the nucleoproteins, designated RNPs, polymerase polypeptides, PA, PB1, and PB2, and non-structural peptides, which are NS1 and NS2. I'll go to the subheadings for this section. Structure. And as you can see there, that's a pretty good picture of it. This represents the influenza A H1N1 virus, but I think it's a good representation for any of the influenza viruses. It's a subtype of influenza A virus that was the most common cause of human influenza in 2009. And then here again, it has the layout of where these different categories can be found on the virus. And then at the bottom there, those are the 2 glycoproteins it's describing and their functions. The hemagglutinin causes red blood cells to clump together and binds the virus to the infected cell. So that's definitely the binding agent for the virus to connect to the cell. And then the neuraminidase are a type of glycoside hydrolase enzyme which help to move the virus particles through the infected cell and assist in budding from the host cell. So obviously that is an integral factor in how the virus leaves the cell and infects other cells. And bear in mind, when the virus is in a cell, mutations are taking place, and some other virulence factor forces are taking place that I'll speak of in a moment. Next slide. This is what the influenza virus actually looks like under the electron microscope. I thought that I would include that. That's a very interesting photograph. Next. Virulence factors. It basically has, I believe they felt that there were 11 genes, but now I believe they have found a recent one. So it's up to 12 genes found on 8 segments of single-strand RNA. So it has a 12-gene, 8-segment, single-strand RNA genome. And genetic replication errors will cause mutation rate, mutation problems, or mutations, which can then be a problem. These are the virulence factors. Another virulence factor is called antigen drift, which is the genetic recombination with the genetic material that's found in the infected cells. It can become mixed with the original viral genetic material, and then when the virus escapes the cell, then it then has a recombined genetic material, and this could give it increased virulence. That's antigenic drift. Antigenic shift is cross-species genetic reassortment, they call it, and well, I have a subsection for that I'll speak of in just a second. Then there's cross-species surface protein characteristics. So let's get to the subheadings. This shows the 8 segmented RNA strands inside, and I think the photograph is mistaken though, because it's showing double stranding there, if I interpret that correctly. These are single strand RNAs divided into 8 segments, and the interesting thing here, and this shows the what was it called? The antigenic shift, cross-species genetic reassortment. Reassortment takes place within the virus, and you can see it here when the virus either in a new host or within or if it comes in the path of other viruses within a host cell, and the genetic material is exposed and then recombined that way, then you have what they call reassortment. It's cross-species lines. It's not just a recombination with the host cell, but it's a recombination of genetic material from other viruses that are infecting at the same time, or what it picks up from the host cell because it was affected earlier. And so now what it has, and as you can see here, these 8 segments, and this is in the this is what is this is one of the virulence factors of the influenza virus, is the fact that it gains strength and virulence from these reassortments, these cross-species reassortments, which then manifest themselves in these 8 different segmented RNA strands. And as you can see here on the right, there's everything from the avian polymerase, avian or human polymerase, avian polymerase, human or swine origin, then swine origin, human or swine origin, and then swine origin for the 8 different segments in this particular virus. And it is this particular virus strain. There would be different reassortments for different strains, and that's what makes the influenza virus so dangerous. Or one of the things is the virulence factor of reassortment. Next slide. Okay, here's the difference between antigenic drift and antigenic shift. The drift is what we've already learned about in the course. It's that gradual change. It's due to point mutations in the H and N, those are the 2 glycoproteins, and occurs in both A and B viruses. Antigenic shift occurs through reassortment of genes, and this is that cross species reassortment of genes. It causes sudden changes. It can spread explosively, has explosive spread, occurs only in influenza A viruses. So on to the next graph. And then this shows here how the influenza virus started from the wild waterfowl in the center, but quickly moves out into other animals, and the animals give it to each other. And as this relates to the humans, we can get it directly from the waterfowl, or we can get it indirectly through swine. And evidently there's a lot of this reassortment that goes on between the waterfowl, the swine, and humans, much more than with other animals, at least in terms of how this affects humans. And as you can see here in the red highlight, we can get the H5N1, the H9N2, the H7N7, and if you notice, like the H5N1 and H7N7 are also in chickens, and the H5N1 is also in swine, and so like I say, the genetic reassortment is what causes, or is one of the most important virulence factors. And as the other graph showed, let's go back to that just for a moment. It causes sudden genetic changes, and it has an explosive spread, it says here. So that's what's so dangerous about the interspecies or cross-species reassortment, genetic reassortment. And then right here, this is it shows how the H1N1 virus evolved, and it says before 1990, and this is the genes that make up the new H1N1 flu virus come from various swine and avian flus, along with the seasonal H3N2 human flu virus. And then before 2000, it says in 1998, the new triple reassortment flu viruses are found in pigs with genes from avian, swine, and human sources. And then it says now, and I'm not precisely sure exactly what they mean by now. I don't know the date of this picture, but it says 6 genes in H1N1 derived from the earlier triple assortment swine viruses, and 2 genes come from Eurasian swine viruses, all mixing in an unknown host. So here again, it shows the significance of the cross-species reassortment, genetic reassortment, and how that is an important virulence factor. Okay, next. Diseases and complications, and as you can see here, we are used to the normal signs and symptoms, but a significant number of people get severely ill, and some even die because of this virus. So it says here it may cause sinus infections, ear infections, bronchitis, pneumonia, epithelial or alveolar hemorrhaging, encephalitis, myocarditis, liver dysfunction, and kidney dysfunction. And it may worsen in those people who already have these chronic conditions. It may worsen bacterial infections, asthma, emphysema, heart disease, liver disorders like hepatitis, kidney disorders, endocrine disorders like diabetes, blood disorders like sickle cell anemia, and immune disorders like HIV/AIDS. So let's look at the pathogenesis. This is a good graph here. Influenza pathogenesis, and I think the significant takeaway here is we pretty much know these factors, but what I found interesting is right here, let me put my finger in here, this is the body's immune response, the T cell response, interferon response, coming down and hopefully fixing or creating a situation which will interfere with the influenza virus at this point here. And if it does, great. Well, then you're cured of the virus. But if it doesn't work, and this is what I find significant, it goes down to these complications, viral complications. And as you can see here, there's viral complications like secondary bacterial infection, primary viral pneumonia, central nervous system and muscle involvement. And it has here as probable pneumonia sources, Staphylococcus aureus and group A streptococci, or cocci, and it has here H. influenzae. So that's my takeaway from here is the if the treatment doesn't work, then there's a high probability of these secondary complications. And this refers to what I was saying earlier. These are CDC estimates of that the influenza virus has resulted in between 9.2 million and 35.6 million illnesses, and between 140,000 and 710,000 hospitalizations, and between 12,000 and 56,000 deaths annually since 2010. That's huge. The problem I have with these figures, though, is look how broad those estimates are on the number of illnesses, 92 or 9.2 million all the way to 35.6 million. That's not very accurate or precise. Same thing with hospitalizations, 140,000 to 710,000, and then for deaths, 12,000 to 56,000. Those are some pretty broad figures. So it's just not very precise. So I don't know exactly what the effect is. We know it's significant, but to what degree, it seems the CDC doesn't even know. And I can understand why. Now, I'll get into this later. Many of these illnesses, hospitalizations, deaths, there's complicating factors. This may or may not be the only illness at the time. It might be making another illness worse. It could be it could have been through an earlier hospitalization than you contracted the influenza virus, or the other way around. You could be in the hospital or a clinic situation with the influenza virus and contract another virus or bacteria. So the situation's quite fluid on this, evidently, to the point where they can't get precise data on the illnesses, hospitalizations, and deaths, as you can see. Next slide. Signs and symptoms, these are the usual. These are what you would expect. Fever, headache, sore joints, coughing, sore throat, nasal congestion, sneezing, irritated eyes, body and muscular aches, chills, fatigue, and sweating. I don't think there's anything unusual there. I did read something about, and many of the sources include vomiting, but they say that actually that's extremely unusual, not a primary symptom of influenza, but strikes most often in the very young. They may vomit, but most adults don't vomit due to this. It's just a side note. Next slide. Transmission. This gets interesting. As I mentioned before, there's the general cross-species direct contact, the bird, swine, human cross-species direct contact, cross-species contact cultures. And what I mean by that is, in the literature, there's a lot of evidence that, I guess especially in certain Asian cultures, Asian locales, the wild water birds, waterfowl, and the pigs and the human beings all live in extremely close proximity with each other. So it facilitates and expedites this rearrangement, this cross-species rearrangement of genetic material that I mentioned earlier. And it's for that reason, and because this normally originates in the Asian areas, is why a lot of the annual testing for the next year's virus is in certain Asian locales, because that's where they figure they're going to find new mutations or any new strains will come out of that in the months leading up to influenza outbreaks here in the States or in Europe. So, and I'll get to more on that later. But evidently, much of the virus mutations and genetic reassortment is due to many of these Asian locales where I say there's close contact between the birds, the swine, and human beings. A significant transmission factor is surface and hand transmission, and well, actually, let's go back one. Human-to-human direct contact transmission via aerosol droplets. That's the most significant transmission force, is the aerosol droplets. And then these droplets can land on a surface, and then you can touch the surface, touch your mouth, nose, become infected that way. So certainly the initial problem is the aerosol droplets, and one of the transmission manners of transmission can be through surface and hand, or directly the droplets can be inhaled into the nose and mouth and cause a direct transmission that way. Portal of entry are the mucous membranes and ciliated epithelial cells of the respiratory tract, and the incubation period is normally 18 to 36 hours. On the subject of aerosol droplets, next slide. This is a great picture showing just what is involved when you sneeze and where those droplets go, how far they go, and like this graph says, just one sneeze may produce 20,000 droplets. So that's significant. And here I'm just showing that these droplets, when they land, or they can infect a surface, and the surface, you may see that there's no difference in the surface. It could be somewhat dry or somewhat moist, and you just don't know. It could be full with viruses, specifically the influenza virus, and then if you touch that surface, or like this shows here, touch someone who's already, their hand already has the virus on it, then you can get it. Then you touch your nose, your mouth, and then that's the portal of entry, and then it goes into your respiratory tract. And I threw in here a photograph of a person washing their hands. That's one of the key prevention factors, is hand hygiene. And here's the portal of entry. It's self-explanatory, pretty much. It's the respiratory tract, and on a close zoom-in version of that, it shows the bronchus, bronchioles, alveola, and it shows the virus going deep into the respiratory tract. It invades the the ciliated epithelial layers, and as it damages those cells, it creates inflammation, and the inflammation it could be part of what I'll talk about in a moment, a cytokine storm, which is can be actually worse than the influenza virus itself. It's the body's immune system essentially attacking itself. So I'll speak about that in a moment. Laboratory diagnosis. Several ways. There's viral culture, which we studied a little bit in the course, involving cultures and the development of the viral plaques on the culture. And then, of course, you do more tests to discover which virus is causing the plaques, so on. There's the RIDTS, Rapid Influenza Diagnostic Testing. This detects viral antigens. It has less accuracy, but can be done in 15 minutes test time. There's immunofluorescence, which we studied a little bit in the course, utilizes both the DFA, which is the direct fluorescent antibody, and the IFA, indirect fluorescent antibody, in staining assays, and has a 2 to 4 hour test time. There's rapid molecular assays, which is the INAA, isothermal nucleic acid amplification. That takes 15 minutes. And there's the RT-PCR, reverse transcription polymerase chain reaction. That takes anywhere from 1 to 8 hours. On the subject of serologic testing, my research showed that it takes specialized tools and training are needed. It's not in this, for use in terms of the influenza virus, it's not accurate, and it's not timely. It takes too much time to get the results. So the CDC actually recommends not using this as a means for laboratory diagnosis. My subheading here, immunofluorescence. I included this picture showing flu-specific B cells, uninfected and infected, and how they would appear with the immunofluorescent technique. Interesting picture. Next. Treatment. Antiviral drugs. These are considered as secondary defense. The primary defense is seasonal vaccination, mostly for people with disease complication risks, like the elderly and those with chronic conditions. Past drugs of choice are more viral resistant, therefore not used anymore, like the amantadine and rimantadine drugs. Present drugs of choice are the neuraminidase, boy, that's a toughie. The neuraminidase inhibitors like oseltamivir, trade name Tamiflu, zanamivir, which is trade name Roenza, and peramivir, which is trade name Rapivab. Also, treatment could include antibiotic drugs for bacterial complications. I have a subclass here, antiviral effectiveness. And as you can see here, this is specifically for the 2016 to 2017 influenza season. These are CDC, this is CDC data, and it says here 100% of the tested influenza for H1N1 viruses were susceptible to the drugs I just mentioned. 100% of the influenza H3N2, same thing. 100% of influenza B viruses were tested and susceptible to those three drugs. High levels of resistance to actually the two drugs that used to be used that are considered obsolete. Still high levels of resistance. This resistance persisted among circulating influenza A viruses, but they're not effective against influenza B viruses. So the CDC says that the two obsolete drugs are basically not recommended for use against influenza at this time. Next slide. And these are the flu prevention tips. Let me see if I can blow this up. And as you can see, they all make sense. Get vaccinated, keep your hands clean, hand hygiene, cover your mouth and nose, don't touch your face. The smile, that refers to, de-stress. Supposedly that'll boost your immune system. Eat healthy, get physical exercise, and stay home if you feel sick. This is prevention, so to stay home after you already feel sick, I don't know if that's a preventive measure, but I would have substituted just the idea of getting plenty of sleep and plenty of rest at all times to keep your system running right. So those are the main prevention measures. And vaccination and containment is a sub area there. Having some difficulty. There we go. And who should get an influenza vaccine? I think I touched upon this before. Basically, it's the elderly, the young to a certain extent, but mostly the elderly, and certainly those who have chronic conditions. From what my research shows, though, essentially everyone should get the vaccine. But you should certainly get it if you're elderly or have chronic conditions, because you want to avoid the complications. And then another factor is containment. So that involves strategies that involve that have to do with coughing and sneezing, how to handle that. And as this shows, basically sneeze or cough into a tissue and then dispose of the tissue. And if you don't have a tissue, cough into your sleeve, the bend of your elbow, your shoulder, something like that, and then wash your hands often. We get back to the hand hygiene concept. So, next. Immunity. Presently, there's no long-term immunity. Seasonal vaccines are multivalent, triple strain. Approximately 100 collection centers worldwide. I mentioned that earlier, that I would speak about that. And a high ratio of those collection centers, like I say, are in the Asian theater, since there seems to be more cohabitation or close habitation of wild waterfowl, pigs, and humans. And that gets back to the genetic reassortment as a virulence factor. So there's approximately 100 collection centers worldwide. Viral strain identifications are made about February of each year. And those are sent from these collection centers to key laboratories throughout the world, so that they can then produce their next season's vaccines. Seasonal vaccines are usually 70 to 90% effective. Viral strain protection is for no more than 3 years, which goes back to the idea that there's no long-term immunity. And simply because you get vaccinated, it's not like getting vaccinated with some of the vaccines for other pathogens. Vaccine for this is relatively short-term immunity. My subcategory is vaccine research. This involves reverse genetics. The viral genome of RNA is converted into DNA, which is then altered in order to remove the genes that cause the viral pathogenicity. The DNA is then converted back to RNA, which produces a new and non-pathogenic version of the original virus. So they're researching this as of now. This is not in production and is not a part of present vaccinations. But this is something they're working on. Hopefully they'll have it in the near future. And from my research, it will be just revolutionary in terms of fighting the seasonal influenza virus. So the next slide. Special comments. Age and cytokine storm, irony. This is what I mentioned before. While children and the elderly are the principal victims of the virus, young adults may be harmed by a cytokine storm immune response. So bear that in mind for just a second. Next section is nosocomial HAI transmission, healthcare associated infection transmission, or sometimes hospital acquired infection transmission, and the infection complications of that. Fear and panic, the pandemic of 1918-1919. From that pandemic, deaths worldwide were 20 million, and deaths in America were 675,000. That's huge, and especially if you consider that we had a lower population at the time. So this is a much higher ratio of the population at the time. And so, like I said, that's why I call it fear of pandemic. We've been through pandemics before. There was about a half a dozen huge ones on record going back into the 1800s. This was the worst one. And it's as simple as a genetic mutation, more than likely not a point mutation, but because those are the slow ones that, if released slowly, our immune system can adapt to, the human immune system. It's the reassortment of genetic material, which is the dangerous part. That's that cross-species genetic reassortment, which can happen rapidly and can express itself in the virus rapidly and be transmitted rapidly, and therefore can cause a pandemic overnight. And that's what they're afraid of. They're not afraid too much of the slow mutations. They're afraid of this reassortment, fast mutation, fast reassortment, and then fast transmission and infection that can come through that cross-species genetic reassortment. Use of masks. And so I mentioned here the socialization, enculturation of this possibility. Seatbelt analogy, which is the fact that seatbelt usage was once very rare and now it's endemic. And that's all just through socialization and enculturation. Now, very few people would even think about driving their car without their seatbelt on. Availability, and it's inexpensive. That's relating to the mass, and possible fashion statement. So I'll get to that under the subheadings here. So first subheading, cytokine storm, and I won't get into too much detail here, but the lower picture says it all. Why complications in young, cytokine storm. It is the systemic expression of a healthy and vigorous immune system resulting in the release of more than 150 inflammatory mediators. Both pro- and anti-inflammatory cytokines are elevated in serum, with lethal interplay of these cytokines is referred to as cytokine storm. The primary contributors to the cytokine storm, and it gives those, and this gives an inappropriate or exaggerated immune response that is caused by rapid proliferation and highly activated T cells or natural killer cells. Bird flu patients die from acute respiratory distress syndrome caused by the cytokine storm and not directly from the virus. And they mentioned bird flu here, but as a whole, the influenza virus does regularly cause a cytokine storm in its victim, which is often lethal. Next slide. This has to do with the nosocomial infections. Approximately 10% of patients admitted to a hospital will contract a nosocomial infection, or an HIA. Approximately 5 to 10% of hospitalizations in Europe and North America result in these kind of infections. In areas such as Latin America, Sub-Saharan Africa, and Asia, it's approximately 40%. That is an amazingly high figure. People who get HAIs usually spend 2.5 times longer in the hospital. And the Centers for Disease Control and Prevention estimate that around 2 million people contract HAIs. Approximately 100,000 of those cases result in death. So that, again, highlights the importance of hand hygiene and containment and the use of masks. I went on the CDC site and on the subject of mask usage, and basically they say that it's only recommended in certain cases, and that you have to use a protocol that would involve hygiene, wipes, and a number of other things to make it effective. But I would say that anything that can help curb the spread of the virus is worth doing. And the reason I brought up seatbelts earlier is it's the same kind of situation, socially, cultural speaking. Nobody used to wear seatbelts, but once it became, once we were educated and we understood why, then we appreciated wearing seatbelts, and I think we would understand why we need to wear masks possibly as a group in public, and so there might be a conversion. I've noticed lately more than ever in my travels that I see people wearing masks. Not a lot, but more than you ever used to see. So I think there's becoming more public awareness, and who knows, maybe generalized public mask usage during the influenza season. It might just be a regular occurrence. So my next slide. These are pictures taken from the 1918-1919 influenza pandemic. Mass deaths, as you can see in the top slide. Mass usage of masks. And even a public service announcement at the bottom suggesting mask usage. So I think that could be a possibility in the future. Generalized mask usage. And then here, I'm just making some points. Mask use is becoming more understood and accepted. Masks, like seatbelts, once rare, may become endemic. Masks are available and inexpensive. General mask use may become popular. And then I put here at the bottom, maybe even fashionable. Because as you can see the man and woman at the bottom of the screen, they both have designer masks out and about in public. That might be something we see much more of in the future. So, next. Here's my references. Questions or comments? I appreciate your time and consideration. Please have a great day. Bye-bye.